So we now begin our second Shaw Prize lecture, which will be delivered by Professor Jeffrey Friedman. Professor Jeffrey Friedman, please.
Well, it's really a tremendous honor to be here on this occasion. I'm immensely grateful and humbled that the committee saw fit to include me in this year's prize. And I'm equally delighted to be here today to be able to tell you about some of the work that we've done and the ways in which we've picked up on the beautiful work that Doug Coleman has done. It's also a wonderful pleasure for me to share this award with Doug, who really is a scientist. Well, today I'm going to pick up the story in a few moments about how the Parabosa experiments actually led to the correct conclusion and what the implications today for that body of work are. But in today's lecture, since the faculty and student body here is quite diverse and not limited to the biological sciences, I thought I'd start with a brief historical perspective about the regulation of energy balance in living organisms and try to make the argument to you that there needed to be a system that regulated body weight in the way that we now know leptin to regulate it. And so with this somewhat historical introduction, I'll tell you about the cloning of the OB gene and how that led to the identification of leptin in a homeostatic system that maintains relative constancy of body weight. And then I'll close largely by telling you about the current clinical implications of this work. And I'll allude very briefly at the end to what some of the new basic science questions that drive us are. I mean, there's a simple question that underlines all of this. And that is, how does nature count calories? Every living organism survives because it can make use of energy in productive ways that allows the organism to perform its functions, to reproduce, and so on. And it stands to reason that every living organism would need some way to monitor those calories, lest in the worst case it run out of the calories and perish. Now, in a sense, every living organism has answered this question. And at a fundamental level, I hope to at least provide some insights into how mammals address this challenge, this biological challenge. Now, in mammals are relatively unique in that they've developed a specialized means for storing energy. Mammals store energy during times of surfeit largely in the form of fat as compared to protein and carbohydrate. And the teleologic explanation is simply that fat is more calorically dense, and so it's a more efficient means for storing energy. And in mammals, this energy is stored as a specialized organ, adipose tissue. Previously, adipose tissue was thought to be a passive reservoir for lipid and for storage, but as I hope convince you later, it's actually quite a more active and dynamic tissue than previously appreciated. Now, in current society, adipose tissue has a bad reputation, and that's because increased adipose tissue is the definition of a clinical condition, obesity, which is a major risk factor for disease and represents a major public health problem in developed countries and is a growing problem in the developing world. And the reason for this is that obesity increases the relative risk of diabetes, hypertension, heart disease, fatty liver, even some forms of cancer. These are collectively known as Syndrome X or the metabolic syndrome. And so for this reason, obesity is considered to be a major public health problem, and adipose tissue is considered to be a nuisance. Now, this is probably, at least in an evolutionary sense, a narrow-minded view of the functions of this organ, because the real role of fat, in a sense, is what I said a moment ago. It's to store energy when calories are freely available so it can be released and provide the sustenance for life when calories are no longer available. And so in mammals in particular, a very elaborate system has been developed that allows the deposition of lipid in adipose tissue and its distribution to a variety of organs when food intake is limited. Now, what I've just described to you is the fact that adipose tissue can be both good and bad. It's good in a sense that it protects you from starvation, but it's bad insofar as it causes an increased risk of a number of diseases that are quite deleterious in the context of evolutionary time. So this might suggest then that there are evolutionary considerations that control the amount of fat that an organism and that a species carries. Let me develop this idea a bit further. As Dr. Coleman mentioned a few moments ago, when we were living as more primitive tribes without domestication, food was often in short supply. We depended on calories from fruits, berries, from hunting, and if there was a drought or some natural disaster of sorts, food wouldn't be available. And so the argument was made in the 1960s by Jim Neal that in this more primitive environment, genes that predispose to the deposition of fat would be advantageous. So in this environment, the so-called hunter-gatherer environment, genes that predispose to the deposition of calories and storage of fat would provide an advantage. And it was further suggested that those same genes in today's time now lead to an increased incidence of obesity when food becomes freely available. Now, you might also imagine that over evolutionary time, and this is probably happening now, in this circumstance, when starvation is no longer a risk, that genes that promote the deposition of fat might actually decrease in terms of their allele frequencies over time. And so what I'm trying to argue here is that there is likely to have been and continue to be balancing selection for body weight in a given environment, balancing the relative risks of starvation against the relative risks of obesity. And what this posits, in a sense, is there ought to be a biological system that can control and regulate the fat and optimize its level so as to balance these two relative risks. To put a slightly finer point on it, it would be easy to believe that an individual who looked like this would have probably a better chance of surviving a famine than with this individual. But on the other hand, this individual would be less likely to suffer a variety of diseases that I just mentioned. And in addition, in ancient times, it's also been argued that phenotypes such as this might increase the risk of being subject to a predator. And so that's yet another reason why there might be balancing selection. So I don't mean to overcomplicate this. It's easy, I think, intuitive for most people to believe that being too thin could create problems and being too obese could create problems, and that there ought to be a mechanism to create a happy medium between the two. And furthermore, that depending on the environment you're in, that line might get drawn in different places. And all of this would suggest that there ought to be a means for controlling the amount of fat that's stored. Now, if we think about that, a mechanism for controlling the amount of fat, this actually means that energy has to be balanced. And balancing the energy equation in vivo, actually inventorying the amount of fat an individual carries, poses a formidable problem. And the reason for that, as I'll explain now, is that the laws of thermodynamics apply to all physical systems, whether they're biological, chemical, or the physical or natural world. It's worth taking a moment now and thinking about where that notion comes from. It has its origins with the work of one of the most brilliant people I've ever read anything by, and that's the French chemist Antoine Lavoisier, who in the 1780s I don't know if it was technically a garage, but at his home did a whole series of experiments that basically created the template for modern chemistry. His day job actually was as a tax collector. This was during the French Revolution, and that could be considered to have been a high-risk occupation. Lavoisier ultimately succumbed to the guillotine, unfortunately. Who knows what he would have done had that not been the case. Now, Lavoisier is most famous, along with Priestley, for the observation that living organisms, that combustion of inanimate systems is analogous to respiration in living organisms with the exchange of oxygen into CO2 and H2O. But he did another experiment along the way building what in a sense was the world's first calorimeter. Here's a schema of that calorimeter. Basically what Lavoisier did is he created a chamber of ice into which he could insert an animal, and he surrounded that chamber with ice and then an insulating material. So if he put that animal inside the chamber, the ice would melt, and he could calculate the amount of heat that was generated over a period of time by measuring simply the amount of water that was produced. What Lavoisier was able to show was that for the amount of oxygen consumed, the amount of heat generated as measured by the water that had melted was identical for living organisms in, say, burning a candle. And this was the first indication that living organisms, in terms of their bioenergetics, obey the laws of the natural world. Actually, in Lavoisier's book, the one I just showed you a moment ago, he describes these instruments in great detail. It's quite extraordinary, the precision and ingenuity that he exercised in building these instruments. This is a picture of the calorimeter he built, and there was a whole host of new observations that were made by building concentric circles and filling them with ice. Actually, he could prevent melting from the outside, and he even developed a port to collect the respiration so that he could now condense the water and get a more accurate measure of the heat that was generated. It's an extraordinary achievement that, as I mentioned a moment ago, gave us the first clue that biological systems are simply obeying the same physical laws. Lavoisier came close to formulating the first law of thermodynamics. He died, as I mentioned a moment ago, prematurely. And so the actual formulation of this was put forward by both, actually by a British physician named Robert Myer. It's often attributed to Joule, but the available evidence would suggest that Robert Myer formulated the first law of thermodynamics with one of the most spectacular observations, clinical observations ever made. What he noticed was that the color of the blood of animals, of sailors, he was a ship surgeon, and he noticed that the color of their blood was different in the tropics than at higher latitudes. And this, of course, had to do with we now know, with the state of hemoglobin in the blood at different temperatures and so on. But somehow, with that clinical observation, Myer worked his way back to the first law of thermodynamics. Because he didn't have any scientific training, he never got the full credit for this and spent his life bitterly trying to explain to people that he had formulated this law first. He was nonetheless unsuccessful, at least of convincing people of that fact. What he had basically said, however, was the following: that in any living system or inanimate system, the amount of energy is constant unless there's a change in the input to the system or the output. So if stored energy in any system is going to change, it's either because there's conservation of energy in or energy out has been altered. Actually, he applied this more to inanimate systems. It was a Prussian surgeon named von Helmholtz who actually formally showed that this same law of thermodynamics applied to living organisms. Now this, in a sense, in a moment I'll explain why this establishes the conundrum that I began by telling you. And that's because if we analogize this to people now, or apply it to people, what we clearly are left with is the following. Energy is stored as fat. If the amount of fat is going to change, it is either because food intake has changed or energy or calories burned has changed. The magnitude of this, however, this operation in living organisms, especially humans, however, is quite profound, because we consume 1 million calories on average per year. 1 million calories, more or less, for the average adult, maybe perhaps a little bit less. Now, it's been well documented in studies all around the world that year on year, weight is remarkably stable in people who are not otherwise dieting or changing their weight or ill. And so what this means, if your weight were stable over the course of a year, is that the amount of calories you consumed as food, 1 million calories, was precisely balanced against 1 million calories burned, and because of that, your fat mass was constant. If we now consider the numerology over longer periods of time, a lifetime of consumption, we consume vast quantities of fat, protein, and carbohydrate. Convert this into waste, heat, and a small amount of work, all the while maintaining a relatively constant weight. In fact, it's been empirically noted that weight tends to increase somewhat with age. The average weight gain is about 10 pounds per decade for the average adult. And so if you do a back-of-the-envelope calculation, what you conclude is that there must be some system balancing energy intake against energy output with about 99.6% precision. Very precise, perhaps even more precise than that. Now, this would suggest there has to be some calorie counting mechanism. Now, I'll point out to you over that same interval, we'll consume tons worth of water and excrete that or perspire in any number of ways, maintaining fluid balance. And this, people generally accept the idea that there are some physiologic mechanisms that maintain fluid balance. But I think for reasons that are more sociological than scientific, people have resisted the idea that there might be some calorie counting mechanism or means for maintaining a caloric inventory. I'll point out to you that despite that the imputed precision of the biological system that sort of operates naturally is 99.6%, this exceeds by several logs the error rate on the food labels you eat for calorie counts. So a group went out and looked at the calorie counts on the food labels you eat, then did calorimetry on that food to figure out how many calories were actually there, and the error rates range between 2 and 85%. So if you were depending on the food labels to guide your caloric intake, you would do logs less well than nature seems to do naturally for each of us, not thinking about our weight. I'll also point out, of course, that every living organism does this, including our mammalian brethren, dogs, cats, monkeys, all maintain their weight relatively stable over a long period of time without thinking about how much they're eating at all. It stands to reason, then, that there might be some biological system that counts calories for us, and that feeding might be represented to a large extent by a basic drive analogous to breathing, drinking water at least, and so on. It's very easy to demonstrate such a homeostatic calorie counting system in animals. Here are two examples of this. In this experiment by Wilson et al., animals are force-fed through what's called the gavage tube. You put a tube into the stomach of an animal, you provide food through the tube, and while you do this, of course, you overfeed the animal by necessity or by force, and the animal gains weight. During that interval, you'll note the animal reduces dramatically its voluntary intake. As soon as you stop the force feeding, the animals lose weight and return to that of a control group, and food intake is reduced until such time as weight returns to that of the control group. It's as if the animal knew that it had gotten fatter and made adjustments to maintain homeostatic control of fat. And the same thing can be seen true for the other direction. If you remove the food or reduce food intake of an animal for a period of time, they will lose weight, but as soon as you restore the food, they'll eat more until such time as their weight returns to that of a control group. It's as if here again, the animal knew that it had gotten thinner and made adjustments in its food intake to maintain homeostatic control of weight. And so this would begin to suggest that there might be a calorie counting mechanism that maintained constancy of weight by regulating how much food you eat and perhaps also how many calories you burn. Now, another way of restating this was put forth in the early part of the 1900s by a number of investigators, including Adolph and others, and it suggested the following: that there must be some means for representing the number of calories that were stored either as fat, protein, or carbohydrate, that these signals were read out somewhere in the brain, and deviation in the levels of stored energy in these compartments would result in compensatory changes in food intake and energy expenditure. Now, I'll point out to you that these three compartments are separate and that there's nothing to exclude the possibility that there are sensing mechanisms for each independent of the other, and clearly glucose sensing is a very powerful mechanism that's evolved to sense glucose levels in the beta cell in the brain, and there might actually be sensing mechanisms for protein or muscle mass that we don't know about yet. I'm obviously going to tell you about a sensing mechanism for fat. Now, partly for the reasons that Dr. Coleman told you, namely the concern about aversive effects, it's been very difficult to use biochemistry to purify these signals. And so as an alternative to that, over the years, my attention was drawn to the very mouse that Doug told you about. This mouse is the Obie mouse. It was described first by George Snell and colleagues, including Margaret Dickey, who Doug mentioned, at the Jackson Lab in 1951. Actually, not listed as an author, but acknowledged on this is a woman named Skippy Lane, who I had the good fortune to meet several times. And I asked Skippy about the early days of this mouse. Skippy told me that when they first saw this mouse after weaning, it was a little plump, and they thought the animal was pregnant, except for three things. It kept getting fatter, it never delivered, and it was male. I like to point out this is why, you can see why George Snell won a Nobel Prize. He correctly concluded that this was a genetically obese mouse and not a pregnant male mouse. Now, this is a really compelling photograph in a way, because the only difference between this animal and this animal is a defect in a single gene. And the result of this defect in some levels is a profound alteration in this calorie counting mechanism. Because I don't think anyone would imagine that this animal is now overeating because of some voluntary wish to become obese. As Doug alluded to, these animals are obese, diabetic, and as I'll come back to when we talk about clinical implications, they also manifest a set of other pleiotropic abnormalities that Doug summarized, that at the time were a source of some confusion, because the abnormalities that were mentioned, hypothermia, infertility, immune abnormalities, pretty much alterations of almost every physiologic system, are not typically characteristic of the obese state. And to my knowledge, at least, many people were skeptical of the fact that this animal would tell us anything about human physiology because of those distinct differences with typical obesity. And I'll try to explain why these abnormalities develop in a moment. First, let me remind you again that Doug also identified a second mutation, DB, and both DB and OB cause an increased food intake, decreased energy expenditure, increased insulin, the net result being massive obesity and diabetes with these other pleiotropic abnormalities. Two mutations with the same phenotype suggest that the genes might be in the same physiologic pathway, and Doug did a classic and historic experiment to amplify that possibility in the form of the Parabos experiments he talked about. And I'll just remind you again, the interpretation of these results were that OB mice are missing a circulating factor that suppresses weight, and that DB mice are missing the receptor for that factor. Now, this was a compelling hypothesis, and it was one that strongly motivated me. I mean, if one believes for even a short moment that there was an opportunity to identify a new hormone based on compelling evidence of this sort, I think most people would grasp that opportunity, and so I had I was in the right place in the right time, in a sense, in a number of respects, because around the time that this was drawn to my attention was drawn to this, a new methodology had been developed that allowed one to clone a mutant gene, not based on any preconceived idea about what its function might be, but rather simply based on a detailed analysis of its position on a genetic map. Now, this methodology is known as positional cloning. I won't go through the technical details of this except to say that it was the implementation of this methodology that Doug finds so boring because it took 8 years. It was boring in its way, but exciting and exhilarating in other ways. And the methodology is a fairly generic one. It was built on the contributions of many people in many places, including Dr. Y.W. Kahn, who described the first means for defining a difference in DNA sequences as an indication of genetic alterations, and also Lap-Chi Choi, who's now the president of Hong Kong University, whose work to clone the cystic fibrosis gene really provided a template for much of what we did. After this 8-year set of boring experiments, the OB gene was identified as a 4.5 kilobase RNA expressed exclusively in adipose tissue. Now, at the time, what we had done is map the OB gene to a small region, took little bits of DNA from that region. That's why it's referred to as 2G7. That was the second microtiter plate, row G, column 7 that I had been characterizing. And that particular piece of DNA saw an RNA only in fat tissue. This is a low-resolution crystal structure of the protein. It encoded a novel 167 amino acid polypeptide with a signal sequence suggesting that it was altered, that it was secreted. And this mutation is defective in the two available alleles of the OB mouse. In the mouse that Snell identified and Doug told you about, there's a stop mutation at codon 105 that ablates protein function. In the other available mutant, the 2J mutant, a retroviral insertion leads RNA to be absent altogether. Now, if you put together the results of the Parabos experiment with the knowledge that there's an RNA expressed in fat, likely expressing a secreted protein, the following hypothesis immediately presents itself. Perhaps that gene, or the OB gene, encodes a secreted protein, which we named leptin, that this protein is secreted by fat as part of a feedback loop that maintains constancy of weight. And the idea would be that this is a negative feedback loop. More fat makes more leptin. If leptin increases, it suppresses food intake, returning weight to the starting point. If fat is lost, leptin level falls. This would be read out and increase food intake again until such time as weight returns to the starting point. Now, for this hypothesis to be true, the following criteria need to be satisfied. The protein should circulate in the blood. Its levels should change with change in nutritional state. And finally, injections of the protein should reduce food intake and body weight in normal and OB mice, but not in DB mice. So we set out next to try to see if these predictions held true. This is the 16 kilodalton protein in normal mouse blood. If you fast a mouse for 3 or 6 days by reducing their food intake, the levels fall. The levels are increased in obese DB mice. This is what you'd expect based on the Parabos experiments. But importantly, if you fast this animal long enough to the point where its fat mass starts to contract, the levels go down. And so in any number of studies in animals and in humans, changes in body weight are associated with parallel changes in plasma leptin level. Now, of course, the key, and I should mention this experiment was done by a wonderful student in the lab, Jeff Hollis. Well, the critical experiment now is what's the bioactivity of the recombinant protein, and we were fortunate here to collaborate with Ketan Gajiwala and Stephen Burley, who are crystallographers and with no prior experience making recombinant refolded proteins, were able to make recombinant leptin that, as I'll show you now, is bioactive. This is a picture of an OB mouse getting saline. This is an OB mouse getting leptin. I think the picture speaks for itself. I'll tell you, though, that the thing I find most remarkable about this photo are the perfectly parallel tails. I remember when Hollis showed me this picture many years ago, I was staring at it and I sort of cross-eyed because something didn't look right. And then I noticed, of course, the tails are perfectly parallel. And I could never get a satisfactory explanation for this. I once went up to Hollis and suggested that maybe he glued the tails. And he denies it to this day. And so you can decide for yourself whether it's more remarkable that the recombinant protein could change the phenotype of the animal so much, or that by chance 2 tails could be perfectly aligned in this manner. Here's the quantitative data from experiments of this sort. If you give the recombinant protein to OB mice, they reduce their food intake dramatically and continue to lose weight over time as compared to control groups. You do the same experiment in DB mice, no effect on food intake, no effect on body weight. This was actually a critical experiment, not just because it fulfilled Doug's predictions explicitly, but because it confirmed that the effect of the hormone was physiologic. Let me explain. If you give a factor to an animal, it could reduce weight because it's physiologic, or it could reduce weight because it made the animal feel sick. This is a so-called aversive effect. Well, if this was an aversive or nonspecific effect, you would expect to have equal effects in both the mutants. The fact that the protein worked in one mutant and not the other told us physiologic, and really, I think, established that this was a novel hormone regulating body weight. There's one other point I'd like to make here, because we also tried biochemical experiments analogous to those that Doug suggested. And it may not be evident here, but you actually don't start to see a measurable decrease in food intake until about 24 hours. And this is with dosing twice a day with much higher levels of protein than you would ever be able to purify out of mouse blood or even out of adipose tissue. And so it became evident as soon as we did this experiment that just the Herculean task of trying to purify this biochemically was just that. It was never going to work. And I think the genetic approach was probably the only one that, even with modern methodology, was likely to identify the hormone. Now, actually, I just mentioned that the effects of the hormone were not evident with respect to food intake till a little bit later. Actually, the first difference that one sees when you inject the hormone is shown on this video. Jeff Hollis came to me excitedly in the morning after he did the experiment. This animal hardly moves at all. It's basically sitting in the corner hoping food appears. This is a leptin-treated animal. This animal got saline. And look, this animal moves around normally. Now, it looks like this animal has already lost weight. That's true. But if you do this experiment, within 6 hours of a single dose, the animal starts to move around. It means this hormone is not just regulating appetite. It's regulating also the voluntary or the involuntary or innate likelihood of moving around. It begins to suggest that many of the things that we believe to be consciously controlled may actually be more hardwired than that. And so this hormone not only changes food intake, but corrects the locomotor retardation of OB mice. Now, the next question, of course, is what about normal animals? In this experiment, wild-type animals are given leptin twice a day. They reduce their food intake for a period of time until about 12% of their body mass is lost. Once 12% of their body mass is lost, the food intake returns to normal. The reason for that is that at that point, all the adipose tissue has been depleted. And once adipose tissue is depleted, the hormone doesn't reduce food intake anymore, as if other signals intervene or supervene once fat is lost and protects the animal from what might be a lethal weight loss. We don't know what that other factor is, but it could very well be there are other hormones coming from other compartments that might get depleted once fat mass has been lost. So that, in other words, at that point in time, reduced food intake would be dangerous. And so mechanisms have evolved to now limit the responsiveness to the hormone. So the hormone, at least as a recombinant factor, cannot lead to the sort of death by starvation you see in the Parabose experiments. And this suggests in some ways that there might be other factors associated with parabosis that lead to death by starvation, because the pure hormone won't do that. And whether that additional factor in parabosis is the same factor, is a factor that resists this compensatory response, is something that we just don't know. Well, these data and similar data from many other groups confirm, in fact, that leptin is a hormonal signal in a feedback loop from fat tissue, regulating food intake and energy expenditure. The next question is, where is the controller? Where does it act? Now, a clue for this came in the form of neuroanatomic studies done in the 1930s by some Yale physiologists, Hetherington and Ranson. And what they were able to show was that if you lesion electrolytically the base of the brain in the hypothalamus, and actually the lesion is not just in the ventromedial hypothalamus, but also the arcuate nucleus, animals become fat. And they become fat with a phenotype that in many ways, not all, but in many ways resembles that of OB and DB mice. Here's a VMH or hypothalamic lesion rat, and here's a normal rat. Now, it turns out that other people had done parabosis experiments in this field besides Doug, the first of which that I know of was done by Romain Hervé in England. And what he had done was to parabose a VMH lesioned or a hypothalamic lesioned rat to a normal rat. What did he find? That when you parabose the rats with a hypothalamic lesion to a normal rat, the lesioned rat gains weight and the normal rat loses weight, just like DB. Well, this suggests a simple hypothesis. The receptor is in the hypothalamus, and you can either lesion the receptor genetically, as in the case of DB mice, or kill the cell that makes it. Either way, you can't respond to the factor, overproduce it secondarily, and the paired animal dies. So this would suggest that leptin acts on the hypothalamus to regulate food intake and body weight. Now, the answer to this question required that you either identify the leptin receptor and and localize it to the hypothalamus, or clone the DB gene and do the same. The first person to clone the receptor was a group led by Lou Tartaglia at Millennium Pharmaceuticals. Our group cloned the DB gene slightly thereafter, and what I'll tell you now is a synthesis of both groups' work because it suggested a conundrum. When Tartaglia cloned the leptin receptor, what he noticed was that it was a family. There were several different forms of cytokine family member receptors. So the leptin receptor is a member of the cytokine family. And what Tartaglia did is took a piece of DNA from the extracellular region, the ligand binding domain, and hybridized it to blots and showed it was expressed ubiquitously, not restricted to the hypothalamus as you might have predicted based on the experiment I just told you. This conundrum was resolved actually by the genetic experiment because the DB mutant that Hummel and Coleman had identified had a very unusual mutation. This mutation only affected one of the different splice variants of the receptor, this B form. All the other forms have short cytoplasmic regions and can't transduce a signal. The B form has a long extracellular region and can transduce a signal in response to ligand. The Calis mutation that Doug identified simply removed this intracellular signal transduction domain. The consequence of having deleted this domain here was a phenotype that resembles that of leptin-deficient mice or mice with a null mutation of the receptor. Now, while probes to the extracellular region were revealed the ubiquitously expressed RNA, a probe from this critical region in the C-terminus gave a very different result, and that is it was highly enriched in the hypothalamus, and moreover, it was expressed in those same nuclei that when lesioned by Hetherington and Ranson, as shown by Hongfei in my lab and Bob Darnell, it's those same hypothalamic regions that when lesioned cause alterations in body weight. So this began to suggest that leptin acts directly on the hypothalamus to regulate food intake and body weight. This was actually a novel suggestion at the time because it's the only peptide hormone that I know of whose primary site of action is the brain. And I'll tell you a bit more about these neural circuits and neural elements in a moment. First, I want to move on to the next step, which is the demonstration that this system is relevant for human physiology. And that demonstration came from the work of Stephen O'Reilly at Cambridge, who identified a child who was of normal weight at birth but began to develop morbid obesity in infancy. The child ate voraciously. His parents reported literally locking the refrigerator to no avail. This child would figure out a way to get into the refrigerator and eat whatever they did. He was already pre-diabetic at the age of 4 when he weighed 90 pounds and had 57% body fat. Now, this child came from a highly inbred pedigree, first cousin marriages a generation or two back, and he had a similarly affected 8-year-old cousin who weighed over 200 pounds. Now, when you have a highly inbred pedigree with an extreme phenotype, the cursory conclusion is that this is a Mendelian or there's a recessive mutation accounting for it. And O'Reilly quickly measured leptin level in this child and found it was absent. So the child was given recombinant leptin, and here's what happened. The child gets thinner despite getting taller over the course of 2 years, is less diabetic, and has a marked reduction in fat. Now, for those people who believe that food intake is purely controlled voluntarily, O'Reilly and Faruqi did an experiment. They put before this child a test meal of about 2,500 calories. This is about the same number of calories that an adult, or 2,000 calories. This is the same number of calories that I would eat, or an adult of my size would eat in a day. This child sat down, ate the entire allotment of calories, and asked for more. They didn't have specified amounts of food available at the time, and so all they know is that the child would have voluntarily eaten at least 2,000 calories. After a few leptin injections, now the food intake of this child returned to that of a similarly aged child. So what was driving appetite in this child was not any alterations of willpower or a toxic environment or the sorts of things that it's generally attributed to. In this case, it was simply a lack of the hormone leptin. And as I showed you before, this is a picture of the child at the age of 8. It turns out that he was constitutively lean, except for the absence of the hormone leptin. I want to stop for a minute and ask you to think about this image. and think about how you would react or what judgments you might make or explanations you might provide if you saw a child like this walking down the street or a similarly affected adult versus a child or adult that looked like this. It turns out, as I'll tell you in a few moments, that at least 10% of morbid human obesity is the result of a Mendelian defect. Now, the majority of those are not in the leptin gene itself. Hormone mutations are rare, and so only about 24 leptin mutations have been identified in humans worldwide. So while they explain or establish a physiologic role for leptin in human, they don't account for most cases of obesity. But a surprising or profound, I think, number of cases of morbid obesity in human are in fact Mendelian. Now, in this child, what's happened is that a mutation in the hormone leads to a failure or a defect in this feedback loop. In a sense, what happens is this child fails to make leptin and, in a sense, perceives starvation. The brain reads this out as the absence of fat tissue altogether. Now, in that context, it's not surprising that the child would eat more and more and more, make more and more fat, but no hormone was ever produced to shut off appetite, and the child keeps getting heavier and heavier. Now, this line of reasoning and many other lines of reasoning have suggested the following mechanism for the homeostatic control of weight. At your stable weight, you have a certain amount of fat and make a certain amount of leptin. Now imagine for a moment that you went on a diet or lost weight for a period of time. Fat mass and leptin level would fall, and a low leptin level of the sort that that child manifested would be read out and cause a stimulus in appetite. And now you would overeat based on a basic drive until such time as your fat mass and leptin level return to the starting point. If, on the other hand, you overate for a period of time, as I have for the last few days, eating higher quality food than this, I should say, fat mass and leptin level would increase, but over time this would be read out and appetite would be suppressed again until such time as fat mass and leptin level returns to the starting point. Now, what this suggests is not that this system regulates weight to the pound or no other factors are involved, but what it does suggest is that there is a biological system that resists weight change in either direction. And so this system provides a means by which an organism can sense their nutritional state and respond to it and maintain weight within a range. Now, while other factors, as I'll explain, such as volition, might seek to drive weight in a different direction than the biological system is driving at, this is nonetheless a biological force that one has to contend with. Now, I'll come back to the implications of leptin for obesity in a moment. I want to digress for a moment and return to these abnormalities I told you about that are manifest in OB mice and are also manifest in these children. Infertility, immune abnormalities, thyroid dysfunction, problems with insulin signaling. All these abnormalities are corrected by leptin treatment, not just the obesity. Now, what this and other lines of research, in part contributed by Jeff Flyer, have suggested is the following. And that is that when fat mass falls and leptin levels fall, there are many adaptive changes, not just in appetite. In a sense, what can be considered is that a low leptin level activates a starvation response, a set of responses that help the organism survive when calories are inadequate. Included on that list are a euthyroid 6 state, altered immune function, reduced insulin signaling, reduced reproductive function. In aggregate, these are a set of responses that seek to conserve calories. And it's all these same abnormalities that ironically are evident in these leptin-deficient children. And so what it suggests is when you don't have leptin, you activate a biological response designed to save energy. Now, this is important in its own right because it tells us something about how changes in nutrition are read out and affect physiology of other systems, but it also has suggested a number of clinical states in which leptin therapy has turned out to be useful. Because a number of scientists around the world ask the following question: are there any pathologic states where there happen to be low leptin level associated with one or more of these abnormalities? Because if there were, then giving leptin back might correct the pathology. Let me explain what I mean. This child was markedly insulin resistant, disproportionately so, and leptin treated it. So let's ask, are there diabetic states associated with low leptin levels? It turns out there are. Lipodystrophy is the congenital or acquired absence of adipose tissue. It's sort of the fat equivalent of muscular dystrophy. Fat doesn't form normally. Because there's no fat, there's less leptin. These patients develop a severe, often intractable insulin resistance, diabetes, and a fatty liver. As first shown in animals by Shimomura, Brown, and Goldstein, and later, as I'll show you, shown by Phil Gordon and others, giving leptin back to these individuals has rather profound effects on their metabolism. Here's a picture of the liver of one such patient before leptin and after leptin, as shown by Oral, Garg, and Gordon. These individuals have a markedly abnormal, life-threatening diabetes with profoundly affected insulin and glucose tolerance tests, again, markedly improved by leptin treatment. So one indication for leptin is certain forms of diabetes. I won't have time to tell you about it, but it turns out leptin has profound anti-diabetic effects in a number of settings, even type 1 diabetes in animals. And so an important area for future research is going to be to try to understand how leptin improves glucose metabolism. A second area where there's low leptin in pathology is a condition known as hypothalamic amenorrhea. This is a condition that suggests, as I'll tell you, that leptin is associated with fertility. Now, this suggestion was actually first made by Fareed Shahab at UCSF and Jeff Flyer at Harvard, but was also suggested by the following observation from O'Reilly. The older of the two cousins with leptin mutations was prepubertal based on FSH and LH levels at the age of 12, despite the fact that, according to her bone age, she should have entered puberty. As soon as they gave the child leptin, she started cycling normally. When they removed the leptin, she stopped. It's as if leptin was somehow required for normal menstruation. Now, this notion fit very well with the hypothesis first put forth by Rose Frisch in the 1960s. Frisch had noticed that heavier girls often enter puberty earlier than thinner girls, and that very thin adult women often stop menstruating. And she concluded from this that there must be a factor from fat required for normal reproductive capacity in women. We now know that that factor is leptin because of studies of it in this condition of hypothalamic amenorrhea. This is the infertility that develops in very thin women, such as gymnasts, ballet dancers, long-distance runners. It actually accounts for a third of female visits to infertility clinics and affects as many as 4 to 8% women of reproductive age. These women are very thin, have low leptin levels, and when you give leptin back, you can restore reproductive function, as shown by Chris Mansouros, and also correct the other abnormalities that develop in these women. So this now provides a link between leptin and reproduction. In the interest of time, I won't tell you about all of these links. I'll simply say what they've established in aggregate is that leptin, that adipose tissue is an endocrine organ, and leptin corrects, essentially serves as a conduit by which changes in nutritional state are read out and lead to compensatory or adaptive changes in probably every other organ system. I think it's intuitive to everyone that changes in nutrition affect other physiologic systems. And what this begins to suggest is that part of that mechanism may be read out through leptin. This has also suggested a host of leptin deficiency states, only some of which I've listed here, whereby leptin treatment may have beneficial effects in human for diabetes, hypothalamic amenorrhea, certain forms of diabetes, and so on. What this doesn't address, however, is the relationship of leptin to obesity in the general population. One way to surmise what its relationship might be is simply to measure levels and ask, in the first case, is obesity typically the result of absent leptin? And the answer is decidedly no. In fact, most obese patients actually have high levels of leptin. What's shown here is percent body fat related to leptin is done by Margarita Maffei and Eric Ravison. And the correlation between percent fat and plasma leptin is very high, and in general, obese patients have high leptin levels. Now remember, leptin normally reduces weight, so if you have high levels of a hormone in the absence of a hormone effect, the cursory conclusion is that this is a hormone resistant state. For some reason, like the DB, analogous to the DB mice, the hormone is not read out properly. There's a secondary increase in weight and leptin level. So the idea here is that there's some block to leptin action at the hypothalamus. This would lead to a transient increase in food intake, increase in fat mass, and an increase in leptin, as if dialing down leptin sensitivity has dialed up weight. I'll point out to you that it's attractive to me, actually, to consider the possibility that, in fact, that's how evolution might work in this system, that by dialing up or down leptin sensitivity, you might change the propensity to obesity or leanness. And this is, I think, an area that's under intense investigation by us and others, that is, what the mechanisms of leptin resistance are. Now, as a pragmatic matter, you might ask, if someone is already leptin resistant and has high endogenous levels of the hormone, what good would it do to give them more of the hormone? Can you overcome the block? And what you might predict from this is probably based on the insulin analogy in type 2 diabetes, sometimes yes and sometimes no. So what's shown here is the first experiment giving leptin to obese patients, and what you'll notice is that there was a dose-dependent reduction in weight, and that some of these patients actually lost a great deal of weight with leptin alone. Now, this was a very small study, and so it turned out that the responder subset was overestimated from this. We now know that about 12% of obese patients lose more than 10% of their weight on leptin monotherapy, and 30% lose more than 5% of the weight. So there's clearly a responder subset that respond to leptin in the context of obesity, but most people don't respond. And so with that as the starting point, there are three ways one could proceed. One could see if there are means of identifying responders in advance and treating them, and it's attractive to postulate that the responder subset can be enriched by measuring leptin levels, and that patients with low leptin levels will respond better. Another possibility would be to try to find a leptin sensitizer analogous to an insulin sensitizer. And then a third possibility, perhaps the most important in time, is to elucidate the molecular basis of leptin resistance. Well, let me start with a brief discussion of leptin sensitizers. And to tell you about that, what I need to remind you of is that body weight is regulated by many factors, not just leptin. In fact, it's thought that food intake and body weight are regulated by short and long-term systems. The long-term system is composed of fat and leptin to a large extent, and it works in parallel with a set of gut and GI tract signals that essentially tell your brain about your recent food intake. So the idea here is that when you eat, factors are elaborated that tell you just eat and shut off your appetite short term. And this short-term system is composed of neural signals as well as a number of other hormones and peptides, insulin, amylin, peptide YY, ghrelin, and others, about which some of you might have heard. Now, what I want to tell you now is that people have long suggested that if you treat only the long-term system, the short-term system will compensate. If you treat only the short-term system, the long-term system will compensate. And so it's been thought that you might want to combine treatments, elements of each system to treat obesity. And now what I'll tell you about is a combination between leptin and a peptide hormone made in beta cells known as amylin. This peptide is sold actually as a clinical product by a company named Amylin Pharmaceuticals. Amylin is co-secreted from beta cells with insulin and has synergistic effects. This is why it's an anti-diabetic treatment on the market. It suppresses glucagon, it suppresses gastric emptying, and it also acts on the brainstem to reduce food intake. And what the people at Amylin noticed was that if you give this alone to patients, they lose durably 5% of their weight. That's pretty good. It's probably not clinically significant enough, but it suggests that this is bioactive. So they now asked, what happens if you combine amylin with leptin? And the data are shown here. The combination leads to about 13% weight loss with a highly significant increase over the monotherapy arms or pure placebo. Somehow, for reasons we don't understand, amylin is sensitizing leptin-resistant animals and humans to leptin treatment. And there are biochemical correlates of this sensitization, but as an empiric matter, it would seem to suggest that this combination might have potential as an anti-obesity agent in human, and that possibility is currently ongoing, being tested in ongoing trials of its safety and efficacy. Now, the final challenge will be to elucidate the molecular basis of leptin resistance, and this is a Herculean challenge in some ways, because it will require that we understand or elucidate the neural circuit that responds to leptin and further determine what's different about the responsivity of that circuit to leptin in the obese state. Now, as I mentioned, leptin is a cytokine family receptor. It transduces a signal through a number of downstream elements, including positive and negative factors. I'll just mention that two factors that shut off the leptin response are a phosphatase, PTP1B, and a suppressor of signaling known as SOX3. You might imagine that these contribute to leptin resistance because they shut off the cellular signal generated by leptin. And indeed, this is supported by the fact that mutations in PTP1B and SOX3 cause leanness and leptin sensitivity. So this suggests that the further delineation of the signal transduction components responding to leptin could lead to new therapeutics. But these aren't simply simple cells. They're cells wired into a network. And the network of neurons that respond to leptin has also been elaborated in recent years in the form of neural pathways that either increase or decrease food intake. The prototype of a pathway that increases food intake expresses the leptin receptor in a peptide NPY. And when these neurons fire, animals and humans eat more. A second pathway expresses leptin receptor in a peptide MSH, which signaling through the MC4G protein-coupled receptor inhibits feeding. Leptin acts by inhibiting these neurons and activating these neurons. So in the absence of leptin, these are maximally active, these are off, and you eat more. With extra leptin, these are maximally active, these are off, and you eat less. And moreover, the system is tuned so that quantitative changes in leptin concentration lead to changes in the balance between these pathways. This pathway has attracted a lot of attention and interest in recent years, in part because mutations in it cause morbid obesity in humans. We now know that about 10% of morbid human obesity is a result of defects either in leptin, its receptor, POMC, or the MC4 receptor, the majority being the MC4 receptor. 10% of morbid human obesity is the result of a Mendelian defect. This is a level of genetic Mendelian loading that exceeds that for any complex trait, and I think would correctly lead people to question, or at least be perhaps less judgmental about why obese people are so. Now, the deep question that devolves from this is, where do these neurons project, and how is food intake regulated at a higher level, because other factors besides leptin regulate it? I'd like to argue that we have a unique opportunity to study a complex behavior because we have a defined signal in the form of leptin, a discrete number of responding cells, and a measurable behavioral output. And so the big question is, what happens in between? Feeding is what's known as a complex motivational behavior. What this means is that many factors influence the likelihood that you'll eat, but none guarantee it. So in addition to leptin, sensory factors influence the likelihood that you'll eat. Emotional factors influence the likelihood that you'll eat, rather such as if you're fearful or depressed. And in humans, volitional factors are relevant, the conscious desire to eat less. And so what has been surmised is that there must be some site in the brain that integrates all these signals and makes a decision, eat or don't eat. Not only do we not know how complex sensory information of this sort is read out in the brain, we don't even know where in the brain it's read out. And so a deep question moving forward will be to understand how all these relevant inputs are sensed and transformed into a complex behavioral decision. I think this question is one that will occupy many scientists for many years, and I don't have any particular insights into the answer to it, although we're trying to address it to the best of our ability. So I'll simply close by telling you that while there are questions that remain about the neural circuit that regulates the behavior, with leptin, I think we have evidence for a clear physiologic circuit that regulates homeostatic control of body weight. It's composed of a classic feedback loop composed of an afferent signal in the form of leptin, integratory centers in the brain, and a set of end organ effects that regulate neuroendocrine state and metabolism. Now, this framework provides the basis for the current work in our laboratory. I won't have time to tell you about it, but I'll just give you a few snapshots of some of the questions we're trying to answer and perhaps save some of the details for future visits. One important question that we don't understand is this. More fat makes more leptin, less fat makes less leptin. It's as if the fat cell knows how fat it is and adjusts leptin production accordingly. Well, that would imply that there's a lipid sensing mechanism in the cells, and we have no idea what the nature of that putative sensing mechanism is. A second question I just raised, how does a single molecule regulate a complex behavior in animals and humans? Where is the decision to eat made? And finally, What is the nature of these leptin-regulated signals that control metabolism, in particular those that improve diabetes, not just in lipodystrophy, but in other forms of diabetes as well? I think these questions will keep us occupied, and as I mentioned, I'd love to have the chance someday to tell you about our ongoing efforts to answer them. I'll simply close now by trying to summarize what it is I've talked about. The cloning of the OB gene and identification of leptin has uncovered a new endocrine system regulating body weight. The system provides a means by which changes in nutritional state regulate all other physiologic systems. There are a growing number of leptin deficiency syndromes that are treatable with leptin replacement. A subset of individuals lose weight with leptin therapy. The majority are leptin resistant, but appear to respond to a leptin-amylin combination. And finally, I think this work shows that there can be a powerful biologic basis for obesity, and that is beginning to, I think, alter the public's perception of the causes of this condition. The advice the obese typically get goes something like this: they should eat less and exercise more. This advice was first offered by Hippocrates more than 2,000 years ago, and I'd like to suggest that perhaps modern science could do better than reiterating a remedy that is no more likely to work today than it was then. It certainly wouldn't work for this child, and I suspect it won't work for the majority of obese people, at least by itself. Rather, I think what we should do is approach this problem the way we do all other medical conditions. Define the molecular components of the physiologic system, understand what's different about the system in the pathologic state, of course understand how environmental and other factors modulate the system, and from this develop rational therapies. And so I think this is at least the charge, I think, for this field for the future. While new therapies may await perhaps more development, I just want to close by telling you that I think in addition to that, I'd like to think we've helped to address the beginnings of a simple question: how to count calories. And one of nature's solutions is create a hormone that's a surrogate for those calories in the form of leptin. So I'd like to thank all of you for your attention and for the great honor that's been bestowed on me here.
Thank you, Professor Friedman, for your interesting findings. And now because Professor Friedman has to catch a flight right after.
Well, let's take a few questions.
Yeah, so we can take 2 to 3 questions.
So the question is, how long would it take to do this if we started today? That's a very depressing question. Probably about, probably one person a year. In fact, we're collaborating with a group from India now that's identified a recessive mutation in rats that causes obesity, and so it goes very fast. The main difference, in my estimation, is really the availability of dense genetic markers. What took so long for us was finding a tightly linked genetic marker, and we had to use some rather elaborate means for identifying them. Now you would simply go to a database of molecular markers and map. Of course, it's also accelerated by the availability of the sequence in a gene inventory, so I think this can go really quickly today. It wasn't the case in the 80s, though.
So, Professor, I have a question. So, is there any leptin receptor expressed in the fat tissue so the leptin could regulate in an autocrine manner?
So, I think that's a really good question. There is clearly leptin receptor at peripheral sites, although in most cases it's the short form. The only exception are cells of the immune system, which have the long form, and there appear to be direct effects on immune cells to modulate immune function in some very interesting ways. The evidence that leptin has effects either autocrine on fat or endocrine on liver, muscle, or other places is not very convincing. I think the probably most precise way to say it is that you can't exclude direct effects on these peripheral tissues, but if anything, they're modulatory. The evidence that leptin acts primarily on the brain include the following. You can replicate all of leptin's effects by giving it at low dose directly into the brain. A brain-specific knockout of the leptin receptor recapitulates the null mutation phenotype, and a transgene expressed only in brain can suppress the DB phenotype in mice. So I think the evidence is that the majority of its activities are on the brain, but I can't exclude an autocrine or endocrine effect elsewhere. So I must admit, I was probably a little glib about this. The question is the hypothalamus the only brain region? No, probably not. There's clear evidence for leptin signaling through reward centers in the nucleus accumbens. And there was even a really interesting report about direct effects on the hippocampus having to do with performance in the forced swim test, which is an indication of depression. And clearly there are emotional sequelae of dieting or starvation that are unpleasant. And it does appear that leptin may have some deeper cognitive readouts of that sort. I mean, one anecdotal set of observations that are consistent with this is that I've spoken to a lot of lipodystrophy patients. And if you talk to them, and admittedly I'm not objective and I may be forcing the issue, but my impression is that what they describe is before leptin treatment is some sort of lack of a sense of well-being, and they all seem to me to at least report an improved sense of well-being when they get leptin. And so I think there are emotional correlates of its responses, and there are probably other brain regions that respond to it and account for that.
So.
Oh, maybe two more questions?
Oh, yeah, sure.
I have a very simple question. So on the evolution, there are some animals they are fat in nature. So let's say the pig. So they have a lot of fat. Is that mean that they have a less amount of leptin?
You know, I don't know. We're going to get right on that one. Well, you know, so all domesticated animals are the products, in a sense, of selective breeding throughout the ages. And in any population, there's natural variation in body weight differences and, of course, in leptin sensitivity, I suspect. And so what was probably done over generations was to select out those pigs who happened to be more obese and more leptin resistant and make more of them and more of them. Now, it's the same thing can be done in a very short period of time. There was a fellow, for example, in Scotland who did the same thing with chickens or other people with rodents. In a very few generations, by selection, you can make animals very fat. Now, I was being glib. I'd bet any amount that the average pig has high leptin levels, but I can't tell you that for a fact.
Would you, as we know that there's a soluble leptin receptor in the serum, could you please comment a little bit on how they interact with these leptin and the signals that giving out?
So there are circulating forms of the leptin receptor that circulate in blood and bind to leptin. There are two sources for it. There are, I didn't really spend any time on it, but some form just lacks a transmembrane domain and gets secreted directly. Another form is probably shed receptor, as shown by Kai Li and others. Now, what we originally had hoped, or believed at least, was this soluble receptor sops leptin up, and that you could inhibit leptin's effects by adding more soluble receptor. While that's an attractive notion, there's no compelling evidence that that's the case. So if you overexpress soluble receptor in liver or elsewhere, you don't get an obese phenotype, and you don't have any attenuation of the leptin effect. And I don't really have a good explanation for why that's the case. I think one thing we want to do, and we're in the process of doing now, is carefully cataloging what proteins leptin circulates bound to. We tried this early on using biochemistry, and the methods weren't so good. But I think with recent developments in mass spec and so on, we can get a much better quantitative readout of how much leptin is actually bound to soluble receptor or other proteins in blood, and that may reveal a fuller answer to your question.
So thank you again, Professor Friedman and Professor Coleman. It's indeed an honor, and I would also like to thank you, audience, for your participation. And before we leave, I would like to finally remind everyone that at 5 p.m. here, Professor Coleman is going to have an informal session, sharing session here. So everyone is welcome to join. Thank you very much, and see you.